Thank you. Thank you, Dennis, for the invitation and the opportunity to meet you all virtually. Great presentation so far and great discussion. My name is Khatereh Motamedchaboki, and like many of you, I'm a proteomics or protein chemist scientist. I used to actually run a proteomics core, and I used to be a director of the NCI Cancer Center Proteomics Core in Sanford Burnham Prepis in La Jolla for 10 years. And I built a whole proteomics core from ground up. And in 2016, the core became one of the Thermo-sponsored core lab. And then I moved because of my family to Bay Area and moved from academic research center into the what they call it product marketing. I was involved in launching Eclipse, Orbitrap Explorers 240, 480, FAMES Pro, and single cell proteomics DIA methods, and you call it max ID methods. I then went to SEER, did plasma proteomics and translational research for 3 years, and now I'm back at Thermo doing global product marketing and translational research. Means like focusing on the large cohort and how our technology and workflows supports bigger studies. And today's talk, actually, I want to show, many of you are running proteomics core, how this technological advancement in mass spectrometry enabling like a data independent acquisition, what we call it DIA, to be the method of the choice for many applications that we haven't taught before. And my colleague, Dr. David Horn, will cover the data analysis part and will as a solution for these bigger data. Get many requests from many people and asking for different things. Sometimes they want to understand the disease states, which proteins or pathway are implicated, how cells are being affected by environment or medications or mutations, or doing the in general biomarker discovery in a smaller scale. And in some opportunities you get access to the larger cohort clinicians or user in of your proteomics facility. And then also drug target identification used to be a really hot topic when I was running a proteomics core, and then protein characterization when they're looking at the PTMs and things like that. And then it's always like which workflow I pick, right, for which type of the experiment, because you try to design a workflow to get the best data possible for each of those scenarios. And what makes proteomics actually important when these guys come and utilize our core, because they want to see what proteins are changing, because sometimes they have transcriptomics data, genomics data, that is kind of a prediction, but they want to confirm and see if they're missing anything at the protein level and post-translational level. And why detecting these ones are important, because they explain the real biology. They're closer to the phenotype, the genome and transcriptome, right? But what we need to see these changes accurately, we need mass spec that are sensitive, or measurement that are sensitive, that they have a really great depth of coverage, and they're consistent, right? The readout or measurement needs to be consistent. And then also one thing that is really important these days, the data completeness, because as more genomics people are coming, transcriptomics are coming to use our facility, there are actually concerns like how complete our data is, because they had to be able to quantify them across the many samples, right? And with that, bottom line is like we need to find the best way to quantify the proteins more accurately to be able to make a conclusion, right? That explain the biology. And for example, like why we sometimes are doing lots of discovery and we lose a sight of how important protein quantification is. And I just have an example of the CRP that most of you have identified from plasma, because it's one of the high abundant protein, but actually measuring the CRP in the plasma of the patient going to a clinic with just congestions, right, it could explain what type of the symptom they are or what type of disease they are and at any stage, right, or any different kind of a viral bacterial infection. And the problem is like just to identify a protein, great, right? But then to be able to quantify the protein, that is the value that you can take into the clinical research or translational research. Because if you go to the, doing any like a protein assay at the clinic, you either see is absolute value or is a range or is a ratio, right? And with the mass spec kind of readout, you can't actually do a discovery that give you those kind of information to transfer to clinical research. And when it comes to the like a MS development or workflow development on any MS, you always try to see as much as possible, right? You optimize the method to see as much as possible protein, but we call it identifiable, right? We have at least one good protein with the good MS2s, right? And then it's like then the list of the protein shrinks to those that you can quantify, and then list of the protein of interest that you're interested in a certain pathway or your customer are interested, but the things is not all of them you can quantify well, right? They either have one peptide, they're missing point across the different, sample, and you cannot quantify them. And when it comes to the method or mass spec development, we try to come with the workflow that for those protein of interest, you move them on the quantifiable bucket, right? And that is like what the thermo teams try to develop in the recent year. We have been doing great in the DDA world. The recent year, the DDA uptake, like DIA uptakes is increasing. People are doing more DIA, more software are developed. Like Diane, I would say it was a breakthrough in the DIA work, because before Diane, you had to generate lots of a spectral library to be able to increase your ID. Now the Diane spectral library match between run all of those and like even Chimerys that my colleague is going to provide more information later today, is enabling the uptake of the DIA method. As I think it was Rick that he mentioned earlier today, even PTM DIA is in uptake. Indeed, I was talking to Evotech, which is the CRO in Germany. They do all their PTM, phosphoubiquitination, everything with DIA now, where they used to have a DDA method before. Means people are using it, and as they're using it, they play with the method, they optimize it. And the beauty of being at the Thermo is we get to work with the scientists in the lab to optimize different method and workflow on, I'll call it our three best instruments that are there for proteomics application. On the left, you see the Orbitraps. I saw great data today on even QEs or, HFX system. The Orbitrap Explorers 480 is one of the newer system, really solid instrument when it comes to the CV quantification. I have been using it for, past 3 years and plus of proteomics, and is a workhorse. And, it supports like metabolomics, proteomics, different application for a multi-user type of the proteomics core. On the center is like a tribrid, the new generation is ASCEND, which practically you have a linear trap. Means like if you need like a higher level of sensitivity of the linear trap, you have that option to run the method on, right? And indeed, there were a lab in Spain that did a DIA using ion trap for low sample input, which was, to me, it was really interesting, but I never tried it myself. And then on the right, you see the Astral, which is like our newest instrument. And it was built for DIA. And because of the speed that it has, and it enables you to get the same coverage and quantity accuracy, but much faster. This one is like on the right, is built for higher throughput methods. Like means if you have like clinician or translational researcher in your your institutes that they have access to large cohort tissue, matched plasma, this instrument is built to enables those large scale studies. And, the things is DIA versus DDA. And, many of us, we were using all DDA, TMT to improve the throughput, PRM to improve the quantification accuracy once we do our, discovery first using DDA. But when the DDA came, many of us, we were skeptical, right? And then as we learn more about the data analysis and also we get our hands on it and we evaluated it, now it's like a really great, benefits of the DIA. And I have listed some of them here, but many of you, I think, have seen it already based on today's presentation. One of the things that is really important for me as a proteomics scientist in translational research is missed data completeness, right? Using the match between run in the DIA or losing the spectral library gas phase fractionation, as some of your colleagues just presented, these ones are the tool that improve your data completeness, right? And then also, since all the peptides are fragmented, like close to the similar M over Z's are fragmented in one window, you have a chance of using, for example, Rudy Abersold bottom-up proteoform detection algorithm to use the spectra of the DIA to look at the proteoform information, right? Some things that we always thought, oh, we need a DDA spectra or MS2 to see if this protein is this peptide from that proteoform or not. There are tools being developed even for DIA nowadays to look at the proteoform from a bottom-up approach like DIA. And that is the value that I see. And this, like a cartoon says as well, that how with the DI acquisition method, you can improve the quantification because of the data completeness. And when it comes to the workflow, right, DIA or DDA, in this case, DIA, is like, there is a whole workflow includes the sample prep, LCMS, and data analysis, right? And my colleague will cover the data analysis, but on the sample prep and the data acquisition, just to say, like running a proteomic score, when you have up to 10 sample, typically is done on the manual way, right? Gels, spot, IP, or things like that. When the project is bigger than that, right? I wouldn't let my technician do more than 20 samples a day because everybody is like, gets tired, like things goes wrong. And to get a good data, especially running a core, you don't want people to resubmit the samples, right? Automation, when you have larger project, is always good because it lets your scientists don't worry about the sample prep and instead focus on the data interpretation, right? And we have an acceleroom that was launched for the TMT workflow. It does a really nice kind of a neat digestion workflows for cell, tissue, plasma. But I'm sure that by now, many of you, if you have access to the large studies, you have automated in some way of automation. It doesn't matter what tool you use. But if you're interested in easy prep kit of the 96 well plate, either automated on Accelerum or Kingfisher, just let us know and we'll be able to give you more information. But this like all depends to your cohort size. How big is it? And what is the value of automation versus manual? And you always get better CV with automation, although I'm really good in pipetting. Sometimes I used to beat a robot, but the thing is I don't know how long I can do that, right? So I'm getting old. Yeah, that's the things that these are the value. You get you can manage your lab resources better, and also reproducibility of the sample prep day in and out and within the weeks and months. And when it comes to the data acquisition, again, it's really important that your goal is ID or your goal is quantification, because you can always tune the method toward one way, right? I saw like Mike was mentioning that he has like how many points per peak on the DIA. I said you can cut it to half and increase your power of identification for discovery, since you're doing the further evaluation or verification at the quant level. And also one thing is really important when you pick the method for DIA or DDA even, is actually the sample throughput. Because you have a lab a certain number of mass spec and you know your lab capacity, right? If you get a big project and your instruments have time, you can always do a little bit longer gradient method, right, to improve the detection and sometimes quantification. Or if you have a higher amount, you can always do like a 1 microgram instead of 200 or 500 nanogram and get better data. These are the adjustments you can do. But in general, that is what the information you have, and you go and optimize your method. And the other part of getting a good DIA method or any kind of workflow is the chromatography. We often underestimate the chromatography and how important it is. And many of you have gone through the pain of changing the column and everything trap. But we have like now a no vanquish columns, which are like a UPAC column, and also PAPSAP column that you could rely on, and you could run longer term. And here is like, for example, 1600 sample injection on a single column. And when it comes to this UPAC column or micropack column, I used to run 5,000 injection on a single 50 centimeter column at 1 microliter flow rate. They're really robust, and you can get a whole cohort of 2,000 sample study done on single column. And many of you, you're doing data analysis, you really appreciate less variation, which is in account for the technical versus the biological variation that you want to capture, right? And this is another kind of evaluation. They say like even different column from this, from different batch of these UPAC column are providing, really good CV when it comes to reproducibility of the identification. And this column is like a chip printing column. Like, it's not a typical packing column. It's really reproducible. Means you can buy like 2 of them, and the data and retention time is really similar using the Vanquish Neo LC system. The other things is, as I say, do you want a method that gives you a better ID or a better quant? Is always a trade-off, right? But nowadays, actually, you don't need to trade off if you stick around a 30-minute, like a middle point, right? A 30-minute DIA method. Because going from 30 to 60 minutes, I will show you in the next slide how much improvement you can get. Is there, but is not that significant. But the quantification accuracy that you get, like moving a 9-minute method to a 30-minute method, is significant, right? Means if you're doing ID, you can always go fast, right? It gives you like 5,000-6,000 protein even. But if you really are looking to see a biological changes, like a subtle changes of 1.2 fold, I recommend not going like a shorter than 20-minute gradient on DIA using this chromatography, right? However, you can change the chromatography and go like a little bit faster, right? And this is like type of the performance you get even with the 200 nanogram of the Hilo Digest, right? DIA method. And it's pretty impressive, right? And this is even on the Ascent system and the Tribrid, and not on the Astral. And the next thing is like application of the FAMES, right? FAMES is like a kind of adding a DMSO in the system. It's like kind of removing the noise, gives you a better, signal to noise. And you can see here on the top, you don't see anything at the single cell of 50 picogram level. But once you put the FAMES, you see the peptide signal coming. Means like if you are interested, or you have some noise going, like even doing the targeted assay, and you want to remove those, like singly charged or like triply charged noise, you can always filter it based on the FAMES voltage and get a better data. Again, one of the things I like is the CV versus accuracy, right? I always say you can measure something with a really good precision, but measure it wrong, right? And that was really important. When I do any DIA, I always look at the CV first, okay? But then I have to evaluate the quantification accuracy. And I really typically like to do the species mix up to look at the whole proteoform changes, protein profile changes, quantification changes across the multi-species to see if my method has a good quantification. And the next step is measuring the statistical power of your workflow, right? How many samples you need to run. And you can do that in a pilot of less than 50 that capture even 1.1 fold change, right? In a large cohort, you can do that. And with that, late last year, our team in the marketing with the application team at the Thermo, we developed a workflow called Velocity DIA, right? Which is an optimized DIA method on all three instruments that I just showed you, Tribred, OEs, and also Astrals. And all of them is with the 50 centimeter UPAC column, right, at the 350 nanometer per minute flow rate, data that was analyzed on the same data analysis like Chimerys, which my colleague will talk to you about it a little bit more. And if you look at it, this data is actually with the Explorers 480. And you see a really good CV of less than 15%, right? With a 30-minute method. And you see you go longer, you get more ID, but at the same time, your CV suffers because then you're deeping into lower abundant proteins, right? Versus like a 30-minute, it has a really nice, what they call it, balance between ID and the quantification on the with the less than 10% CV, which is really amazing to me. And for the 9-minute, we have two types of methods. One is called max quant, and then it's a max ID, which is one toward better CV and better quantification accuracy. The other one is like toward the max identification. And based on the application of what you want to do, are you doing IPMS, like you're enriching something like a, and identifying it, or you can use max ID methods. But if you really want to do a cohort study or wild type mutation treatment study and you want to see small changes, I will go with the more like a quantitative method. And the 30 method looks like a really good balance when it comes to CV and coverage. The next question is how accurate this quantification is. Okay, I have a really good 10% CV. And the next, there's a similar performance for Ascent that is here, a 9-minute method and also a 60-minute and 30-minute method. Accuracy is still less than 15% CV, which is amazing. And then is like, okay, how the accuracy performs. And this is a 3-species experiment that we typically do at Thermo to evaluate any method. And in this case, this is like a yeast and E. coli changing at the 4-fold and also a 0.5-fold change. We want to see how at the big right and then small changes we can capture with this method. And then we measure it using all these 3 methods, like 30-minute, 60-minute, and 9-minute method. And you see how great the identification is. And this is using the SpectroNaut direct DIA, and we also have data on the Chimerys. And the good things with the SpectroNaut or Diane is they do have the LFQ bench in them, which is like they can do MS2 quant readout or MS1 quant, and their MS2 quant is really good and optimized for label-free quantification, especially for DIA. And my colleague David will walk you through the Chimerys as well. And if you look at it, like going from 30-minute to 60-minute, your quantification, kind of improves, right? But for majority of the protein on average, you have a really good quantification using all methods. And on the 9 minutes, you see your quantification start, right, suffering with the max ID method. However, you can adjust your method to get the max quant, like more like point per peak, right, and get the average quantification really close to the expected range. And the number of identifications still is really interesting. The same performance we have seen with a little bit higher IDs on the Ascent, right, instrument. Again, Ascent is really perfect for TMT approach, but you can also do DIA on any orbit trap that you want. It's the same thing on the 99-minute method on the same profile that you see, better with the max quant method workflow versus max ID method. And I want to end this presentation with Astral, because this box, I call it MS box, was built for DIA performance, and the speed that it has is unmatched. The depth of the coverage it gets you, it's just unmatched. You cannot get it on any other mass spec. And believe me, for the past 3 years, I have tried many different vendors' mass spec to get the best DIA method for plasma proteomics, and the Orbitraps keeps beating every mass spec so far. And if you look at this data is not even generated by Thermo. It's generated by another core facility in Denmark, Jasper Olsen's lab. And you see, like, they have identified with 1 microgram, if you don't have a sample limitation, with a 24 sample a day at 12,000 from HeLa. This is like close to what transcriptomics says that proteins could be expressed in a single HeLa cell, right? Means you are getting close to the 100% coverage for a HeLa, right? Which is like totally not seen before, right? And this is in 1 hour of analysis time, right? End-to-end analysis time. And then if you have lower sample input, like a 200 nanogram, and you can still get like close to 11,000 or 10,000 with the even much higher throughput, right? 60 or 100. Means like this type of things, like it actually improves your performance. It improves your lab capacity, right? Instead of like going and getting a project of like a 6 condition with 5 replicate, you can now pitch to your institute to actually think a little bit bigger, right? And do the discovery in more powered way than like doing the discovery in less powered, and then you have to evaluate it with a larger cohort, verifications, right? Now you can do your discovery in really well powered, bigger study, then your biomarker has a better chance of succeeding in downstream verification, right? Hopefully together we can change the way like people do discovery proteomics, even from cell, tissue, or plasma, it doesn't matter. And then the next thing this customer has done, and that was like an early access customer, it was to Astral, was the same thing, E. coli and yeast spiking in human background, and they see the similar performance, right, measured versus things at the twofold and different, like from a small to twofold, like 0.2-fold and also a 2-fold change. And the beauty is, like, if you filter the data based on CV, right, look at the number of ID. The number of ID doesn't change that much, right? It's like 2 to 3,000 protein you missed, and the rest of them are within a really good CV. And the quantification, look at it, it's like 0.25 versus 0.21 and 2.04 versus 2-fold change, right? It's really solid for these small changes that you want to see. And what does it mean, right? Oh, I'm identifying more with the good CV. Like, you're digging deeper into proteome. You're identifying proteins at the area that you were not identifying before. And if you identify and quantify them with a good CV, right, means you can quantitatively measure them, right? And what those proteins are, and if you look at it, some of them actually are in really low copy, right, expression level in the cell, and it's like and then you can even see them even at that low copy with a really like 1.64 versus 2-fold ratio change, right? Really good, accurate. And then the best thing is, like, okay, I have DIA, right? I have evaluated in the cell and tissue, and what is next, right? because this box can do so many DIA in a day, like a 48, like with the 30-minute method, what can I do? And one application of it is actually plasma proteomics. We have evaluated with the neat digestion, like easy prep neat digestion, 96-well plate, and also with the Sears protograph system, which use a nanotechnology for sampling proteome in the plasma or biofluid. And even in like a 48-minute total run time, right, you're identifying 6,000 protein from a plasma, which this is, and even like close to 1,000 sample, right? And this is microflow, it's not even nanoflow. In nanoflow, you get close to 1,000 to 1,100 based on Geneva Nike's data that was presented at ASMS. But this is microflow, 1 microliter per minute, and you're seeing really great, performance. And that goes with, the depth of the coverage you get, the CV is within 12%, right? And the next one, I don't have much time, but I just want to say you can do the same DIA performance and be quantitative at the single cell level. There are many ways to do single cells, and then DIA is one. And if you know about Thermo is promoting the Sullivan for the cell sorting and digestion. And also we have like a high throughput 50 sample per day workflow on the EvoSep that goes on the Astral with more than 4,000 protein from a single pillar cell. And then you have option to do the library based or direct DIA, but you always get 5,000 or more than that using a single cell, or even more than 7,000 protein at the 250 picogram HeLa digest. There are like many DIA methods for single cell that they have explored wide window and narrow window, and they're published here, and we are really open to share this publication with you guys to see if you can further improve from this baseline method that we can transfer to your core to evaluate in your own hand, and then further optimize it. And these few of these customers have done that. They have started with our method and built on top of it and improved it further, either with different chromatography or mass spec. And I just wanted to end it with, there's over 1,000 publications just last year on Orbitrap to do DIA, and many of them are actually not on Astral or OE because these instruments are new in the DIA, and they're actually on QE, HF or Plus or things like that. Means like people are doing and publishing greatly using Orbitrap in DIA, and with like these 3 instruments that I just showed the performance, the new publications are coming, right? And with that, I just want to say that we are actually getting close to the full proteome coverage using this DIA method with 1 microgram and at the throughput and scale that was not possible before, right? And then FAMES provides sensitivity, and also the throughput of 20, 36, and 80 sample per day is not possible, right? And then you not only get a good depth with DIA, but you also get a quantification accuracy. And I want to also suggest you look at the bioarchive for Orbitrap Explorers publication. Recently, there was a publication, an 1,800 sample study using Explorers 480 for DIA, and it's a 30-minute method. And they have been able to complete, I think, close to 2,000 sample in 11 weeks. Means that you can now go and pitch this to your institute's researcher to say like your lab has this technology to be able to support the research. And I'll be here at the end of the talk to actually get your questions. If you have any question about this workflow and how you can transfer this method as a baseline method to your lab. And I give the presentation ball to my colleague, David. Thank you.
Thank you. Can you hear me? Actually, while it's fresh in mind, do you want to answer a couple questions now while I share my screen?
If you have any question until David share the slide, I'm here.
Yes.
Otherwise,
yeah, nice talk. So that slide where you mentioned combining the seer technology with plasmid proteomics. What volume of serum do you use for those studies? Do you need more serum for those?
No, the things that the seer workflow you can do from 20 microliter of the plasma up to 250. And then adding the 250, like a higher volume of the plasma, actually allows you to enrich the lower abundant protein a little bit more, right? To get on the mass spec detection limit. But you can use it from 20 microliter per sample.
Okay, thank you very much.
Sure.
Okay, maybe I'll just jump into my presentation. I guess I have 15 minutes left to present this, and actually I have more slides than I can probably go through. So apologies in advance if I skim over some details. Also, I'm not going to share my camera, at least at the beginning, because I have some bandwidth issues. So hopefully you can hear me loud and clear.
Dave, can you hear me?
Yeah.
Yeah, we don't have a 1 o'clock hard cutoff. So, if you want to go a few minutes longer, we got started with lunch a little bit late. So,
okay, good to know. Okay, then I will try to keep a slow and measured pace then. All right. So just a couple of things I want to cover in this presentation, and Khatereh mentioned Chimerys for DA quantification. I'm actually going to go not just discuss Chimerys, but also just Proteome Discover in general. So just a quick introduction. I am the product manager of the Proteome Discover software. I've been at ThermoFisher for 15 years. My background's actually in proteoform analysis, top-down proteomics back in grad school, and I've been generally involved with software development throughout my career. And I've been the product manager of Proteome Discover for the last about 8-9 years or so. Did some biopharma stuff before that. So the first half of the presentation, we'll just give a kind of a broad overview of Proteome Discover and just a little bit of detail about how Chimerys is now supporting DI quantification. And then I want to make a change for the second half, talk about our new Ardia platform. So as Khatereh was talking about, for DIA, we're talking about 80 samples per day, 40 samples per day. When you run things that fast, you tend to accumulate lots of files very quickly. And so I want to show at least a little bit about how the Ardia platform helps you manage that data, manage data with projects, and also a little bit about how it helps you keep track of your instrumentation. So Proteome Discover software, it's actually a pretty substantial software package. It's been around for about 15 years. And the main reason for this software package and the reason why it's developed its specific way is to handle the very kind of complex data that our instruments can produce. So if you think about our tribrids can do ETD, ETHD, CID, HCD, UVPD. We have MS2, we have MS3 acquisition methods. And we need our proteomic software to be able to analyze all of those. So from the very beginning, the architecture underneath Proteome Discover is called this client-server-based workflow processing system, which has a collection of nodes that you can draw on a workspace that allows you to do filtering by MS level, filtering by fragmentation level, fragmentation type, but also allows us to deploy new algorithms into the software more quickly. So Proteome Discover is not a search engine. Proteome Discover is a system that allows us to put search engines in it, but also filters, PTM analysis, FDR algorithms, and so forth. So it's really expanded dramatically over the 15 years that it's been out. Partway through the Proteome Discover process, around PD 2.0, we added the study management feature as customers' data sets got larger and larger, keeping track of all the data files, assigning meaning to those data files. So that data files, this disease sample, this other data file is a control. And especially when we're thinking about TMT quantification, where we have multiplex quantification, mapping a quant channel to a study factor is even more important. This allows us then to do statistical analysis, replicates, better visualization, and we produce the results not as a mass spectrometry by file, but actually if you use a study management, it'll do it by sample. So you can so that it makes it easier for non-mass spectrometers to interpret the results. We've also had services for biological annotation. So with the proteins, we'll add gene ontology terms, pathways, protein families, can do things like overrepresentation or enrichment analysis as well. A lot of work was put into the user interface to link the views between the protein list, the peptide list, PSMs, quant values, pathways, and also to the graphical tools. So the volcano plots, chromatograms, and so forth, and you'll see some screenshots of that later. And then finally, in the last releases, we've been trying to put more effort into using AI-based tools to increase confidence, increase the amount of IDs that you can get from a different data set. So there's a lot of words here. You'll get these slides later. You can look into it. But what I showed before is that our instruments support many different kinds of workflows. So we have TMT Quan, we have MS1 based label-free Quan, we have Sci-like type precursor Quan, now DIA, which I'll go into more detail in a bit. We need to support, we can support a bunch of different search engines, Sequest, Chimerys, Comet, MS Amanda. We work with third parties to bring in mascot nodes, MS Fragger nodes, top-down nodes, cross-linking nodes into the software. And so the tools that you have in Proteome Discover spans a wide variety of different kinds of workflows. And we update the visualization to keep up with that. And Proteome Discover is being used for all sorts of different kinds of proteomics applications. If you do a search for Proteome Discover in the literature, see it's used for single cell, immunopeptidomics, interactomics, metaproteomics, phosphoproteomics, glycoproteomics. It's being used for many, many different kinds of things. But I don't have time to go into all the details about all the different workflows, but because there's a DIA focused in this meeting, I'll just talk about how Chimerys and Infirus are used to support DIA quantification. And just to note that DIA quantification was released as part of Chimerys with the PD 3.1 release that was last September. So previous versions of Proteome Discover do not have support for DIA workflows. So to give proper attribution, the team that we partner with, it's a company called MSAID, and they are the developers of these two algorithms, Infirus and Chimerys. They're very experienced mass spectrometrists. The founder has come out of Bernhard Kuster's group at Technical University of Munich. And they actually were involved with the original PROSIT algorithm years ago, which is a deep learning tool for spectral prediction. So they've helped us implement two different algorithms into Proteome Discover. One is called Infirus, which is kind of like PROSIT. So it's a deep learning based algorithm that you input a peptide sequence, modification, collision energy, charge state, and out the other end comes a predicted MS spectrum. And you can use that in one case I'll show in the next slide, but there's actually many different applications I don't have the chance to go into here. And then there's Chimerys, and I will go into more detail about how Chimerys works. And Chimerys is built upon the Infirus algorithm, and it's used at a spectrum by spectrum level to substantially increase the number of IDs we can get per spectrum. So here's one application of Infirus. So this is a view that's taken right out of PD. This is the annotated MS spectrum. So the spectrum on the upper half are the experimental mass spectrum, the peaks that were detected, and the annotations from Proteome Discover we see for that. This is a phosphopeptide. The spectrum on the bottom part is predicted on the fly in the software and presented in the mirror plot. So when you click on a PSM or a peptide in PD, if the peptide is supported by the prediction algorithm, so that right now it's TMT, TMT Pro, phospho, oxidized methionine, carbaminomethyl the number of modifications we support will go up in the future. But if it's predictable, it will show up in the mirror plot. So if you're trying to validate an MS spectrum, this Infirus prediction will really help you. In this case, you can see that this phosphopeptide that we measured is predicted correctly measured by the search engine as from the prediction down below. So if you have any kind of collaborators who want to, are you sure that that peptide is correct? This Infirus prediction will definitely help you convince your collaborator that this is what the peptide should look like. Now, Chimerys is named Chimerys because it is meant to handle this problem with chimeric spectra. So a chimeric spectrum looks like this. So the left is the MS1 data for a complex mixture, wide isolation window, actually something like you would see in DIA, but you also see in DDA. And then all of those peptides within the isolation window get isolated and fragmented, get together, and produces the spectrum on the right. And that spectrum on the right has at least 10 peptides being isolated and fragmented together. Typical search engines don't do a very good job with the spectrum on the right. So if we take Sequest, which is our default search engine in PD, we might be able to pull 2 or 3 of those peptides, but not 10. The Chimerys algorithm is designed to get up to all 10 of those at the same time in that spectrum. So the way that the Chimerys algorithm works, so right now we're seeing the experimental spectrum on the bottom half of this mirror plot, is it's using that Infirus algorithm when it's trying to match a peptide to a spectrum. So it's always doing predicted fragments. It's closer to like a library spectrum match than it is to like a standard stick plot you get from like a sequence search, for example. Now, this doesn't use spectral libraries. It's always library free. I know that Khatereh talked about library versus library free. This always takes in a FASTA file as input. So then once it finds out what peptides or what mass could fit that spectrum, it will try to use as few peptides as possible to reconstruct the original spectrum, but also narrow down by retention time. So there's another deep learning tool built into Chimerys that does a retention time prediction. So it's only going to try to match peptides within the retention time range that's expected to that specific spectrum. And it actually does target decoy as well. So at the end of Chimerys, you get a list of peptide IDs, but you also get an FDR, a Q value associated with each of those IDs. And it's very carefully FDR controlled. So 1% FDR coming from Chimerys really does mean 1% FDR. So for DDA data, which is the original release and back in PD 3.0 about a year and a half ago, Chimerys showed its value over sequence by simply, for complex mixtures, dramatically increasing the number of IDs you can get from that data set. So with the previous versions of PD before Chimerys that use sequest and Chimerys afterwards, it's a dramatic improvement about, in this case, 30% more proteins, almost double the number of peptides, 4 times as many PSMs. But when we were showing this to customers, they say, well, the data looks awful lot like DIA data. Does it work for DIA data? In PD 3.0, it didn't, but in PD 3.1, it does. And so PD 3.1, it goes one step further. It does ID spectrum by spectrum based as before. So there's no calculation of chromatograms at the fragment level. It purely goes spectrum 1, tries to get an ID. When the next cycle comes around, it has to identify that peptide again. When the next DI cycle comes around, it identifies again. But we do one more step further, is that when we're doing the matching, we're not just identifying, but we're also quantifying the relative amount of that peptide in that spectrum. And at the end, Chimerys then takes the quan values for that peptide in successive spectra, calculates an area under the curve for that peptide and produces it in the final results. And we have a node in the results called the Frabenius quantifier that takes the quan values for each of the precursors, the peptides by charge state, rolls it up to the unique peptide and the protein level, exactly like we do for other quan values, methods for in PD. So if you've used PD before, you use it for TMT, you use it for label-free quan, the way it looks and feels and acts for DIA analysis is almost exactly the same. So the way we present the results for Chimerys results in the final output, this is an output for a PD result with the proteins linked to the unique peptides for that protein, abundance ratios calculated using that study management for the different samples. Just like so you get ratios, you get quan values, get variabilities, and you get chromatographic profiles for each of these peptides, for each charge state, for each sample color-coded by the study factor. So I'm not going to go into, so there's only a few minutes left here, so I can't go into too much more detail. But one last thing I want to show on the Chimerys side is, how it interacts with the statistical analysis is the same as before. So we have volcano plots. You can double click on any one of these points. It'll show the protein highlighted on the proteins list and any kind of associated tables. You can right click any of these quadrants, select these as up or down regulated proteins, and you can do things like enrichment analysis that for all the proteins that are differentially expressed, what gene ontology terms, what pathway terms are significantly over or underrepresented. So after that very brief survey of PD, and I'll be happy to answer questions about any aspect of PD if we have time. Just want to jump into the Ardia platform. So I mentioned at the beginning, DIA acquisition, especially for the Orbitrap ASO mass spectrometer, can acquire a lot of DIA data sets very quickly. And now you start to deal with, you're having dozens, hundreds, thousands of files that you have to manage. And data management, of course, is a problem once you start having increasing amounts of data. So this Ardia platform was originally launched about a year and a half ago, and we released this version of the Ardia platform that connects to Proteome Discoverer at ASMS last year. And the Ardia platform is a data management system, an instrument management system with some applications that you can install into your lab. So the way that it's deployed into the lab right now is as a server. We actually have 3 different servers for different applications. There's a lower powered server for HPLC-based, more routine applications. There's one for biopharma, and there's an advanced server, which is meant for proteomics. So you can see that this server is powerful. It's got a really powerful GPU. The reason for that is we actually have a local version of Chimerys running on that server. The version of Chimerys that pretty much everybody's using right now is on the cloud. And so most people have been sending their data up to the cloud, running the data on the cloud. Chimerys needs a very specific type of computing setup for the deep learning. And so it's either on the cloud right now or this local server version, which is much faster, more powerful, can run more Chimerys runs at a time. You can also see it has 128 terabyte RAID storage. The effective storage is more like 80 to 90 terabytes of data. And I'll show where it kind of where it fits into the lab. So the first place, you have an Ardia server on that system is sitting in the lab. You can connect it to Chromeleon Excalibur. So once you acquire data, you can have it automatically upload to the Ardia server for storage. Then you can access that data on the Ardia server through our application. So if you want to load that data in Chromeleon, load that data into Biopharma Finder, and now Proteome Discover, you can load those into studies or analyses and run them. Even though the raw files are remote, they'll get pulled down locally, run locally, and then the results are stored there. But then you can save the results back onto the Ardia server as well. Now, the Ardia server, it's like a Linux box that has a full application system on there. So you can log into the Ardia system from pretty much anywhere within your network using a web browser, and you have access to these applications, the Data Explorer. There's a data review application that works a lot like Freestyle, and an instrument management application that I don't really have enough time to go into. But once you register your hardware to that instrument management system, LCs, GCs, mass spectrometer systems, it'll show up there and have a real-time display of what those systems are doing at that time. Now, there's Data Explorer application, kind of like OneDrive. So this is where you can upload data, you can download data, you can control access to different parts of the data system. So if you want to share a result with a collaborator, you can give them access to that one folder, and that's the only thing they're going to see on Ardia. So you can use it kind of like OneDrive to share data. And if you have access to outside the firewall, you can put something out on the internet. You can share data files and information out to collaborators outside. So the one thing I just want to show one screenshot for the instrument management application. There's an instrument scheduling application in data management. So I know that if you have multiple instruments, multiple LC systems, multiple mass spectrometers, making sure that they're up and running all the time and they're being properly used is a logistical challenge. And this application, one of the many different parts of the instruments application, will help you do that. So you can schedule an application for that instrument at that time. And you can see on the calendar who scheduled it and then what application that they're gonna use for that specific device. How much time do I have left? I just want to know how much detail I'm still allowed to go into.
Not to be too quantitative about it, but I think we started 8 minutes late. So
Okay.
108.
I got you. Very achievable. All right, so the last thing I want to show with the Ardia system is that Ardia and PD together provide more value than what I showed you with PD before, and what Ardia I just showed you now is that there's kind of an emergent set of properties that you get when you have this setup called Proteome Discover powered by Ardia. So I'll go into some of the features you get, but what you can do if you have this setup, so you have an Ardia server, you have a PD data analysis computer, and you have an Excalibur system, is that you can upload your data from Excalibur into Ardia with a Proteome Discover data analysis method, and it'll immediately start analyzing the data post-acquisition. So I'll show you how that works just in a moment in a few slides. Also, though, as I mentioned, this specific Ardia system has Chimerys on it. So there's a different node in Proteome Discover. There's a node that says Chimerys that sends data to the cloud. There's another node that says Chimerys on Ardia server, which keeps all your data inside the organization. And the Ardia version here has at least 10 to 20 times the capacity of any cloud version of Chimerys. So you can throw all sorts of data at this Chimerys, keeps all your data local. There's no security issues with sending anything out into the cloud. So when you connect Proteome Discover itself to the Ardia platform, you get these, you type in a license key and you get a new set of features that you would not get if you installed Proteome Discover by itself. So one, you can see in the upper right, is there's a new add remote files, which allows you to load files from Ardia server into a study. So if you've acquired all your data and uploaded Ardia, or if you just uploaded through Ardia through that Data Explorer application, all your data is centrally located. Proteome Discover has no problems. You can grab it, put it into your studies, and analyze it as normal. The only thing it's going to do is it's going to temporarily down those files onto the data analysis PC before it runs, and then it'll delete those from that PC. There's also another button where you can upload your result files to the Ardia server. So if you want to share a result with a collaborator, you could copy the PD result file over, give them access to the PD viewer. But you could also actually just share it through Ardia server and, like OneDrive, send a link to it, and then they can open it up directly in Proteome Discover, that viewer, by using this add remote files. So the Chimerys and Ardia server I just mentioned, and then the automation I'll show just in a bit here. So as I mentioned, the way the automation works, so this is Orbitrap Astral mass spectrometer. It could be any thermal mass spectrometer. Doesn't have to just be the Astral, it's just an example. So the way the automation will work, as I showed, is the data will get uploaded to Chimerys, to Ardia. Ardia will then send it over to the PD data analysis PC, which will then run the workflow and then save the result back on that Chimerys server. And it'll happen one by one immediately post-acquisition after you acquired each data file. Now, if you have this set up, this is what the Excalibur sequence will look like. So I know in this case, I have an Ardia server in the lab. I have a Proteome Discover PC that's been designated for automation. I get this new option at the top bar here where it says Ardia platform processing with Proteome Discover software. As soon as you click that, it will communicate through the Ardia system what data analysis methods that you have available on that Proteome Discover installation, what quant methods you have available, and then you set where on the Ardia server where you want to save the result. So the processing methods are actually what we call analyses. It's a combination of a processing and a consensus workflow for those of you who've used PD. And so that will give you the complete results onto the Ardia server once that data has been acquired and the data is analyzed. So the way it kind of works looks like this. So the current practice with Proteome Discover is, say you have 50 files or 100 sample DI run that you want to do. You wait till your data has been fully acquired, and then you create a study in Proteome Discover, and you run all those 50 or 100 files all in one large analysis and produce what we call a multi-consensus report. It's a PD result file. If you use the Ardia automation, it will analyze the data immediately after acquisition, file by file, and produce each one of these individual result files per file on the Ardia server. And then you run in Proteome Discover, you take all the individual results together, run what we call a multi-consensus run, which groups all the results together into a larger run and does the comparisons across files and so forth. So the advantage of doing the automation is just time to result. So you only need to start that multi-consensus run after the analysis of the last acquired file for that study has been finished, and you'll get to your result a lot faster. Just one note about the automation is it's not only DIA. You can run pretty much any kind of workflow in Proteome Discover with the automation. If you can create that method in Proteome Discover, you could automate it this way. And just one final thing, I know we're out of time, is that there's no limit on the Ardia server to the number of connections. So any Ardia server can any number of PC clients and then any number of instruments connecting to it. The only thing that once you put so many instruments running through that automation together, at some point, it could end up with a backup in the queue of analyses to run. But that's a couple Orbitrap Astrals when that starts to happen, or up to like 10 hybrids and tribrids. So it can keep up with a lot of instruments. And if it does get backed up, it'll just run through the queue. You don't lose any data. The file's sitting in Ardia, and it's just waiting for PD2 analysis. So if you stop the instruments, it'll just completely finish all the analyses and eventually get done. One final note is that you can, within the Instruments app, you can do these called instrument bundles, where you can send information about your instruments to service, so we make it easy to do that. And also, Ardia has ways to back up and archive files, so as that Ardia server, even with the 80, 90 terabytes of storage, it'll eventually get full, and you need to find a place to back up or archive your files to somewhere else within your network. So one final point So not everyone, of course, uses Proteome Discover. I heard Diane, Spectronaut, Mascot, MaxQuant. There's all these other applications, of course, that all of you might want to use. So only our native applications are now connecting to Ardia using these native UIs. But we do have another tool called the Ardia Platform Data Sync that you can install on any desktop PC where you want to do data analysis. And once you install that, you can target a folder on multiple folders on the Ardia platform server, and it'll download all the raw files from that folder to your desktop PC. So you can have the automation upload those files to Ardia, but you still want to analyze it using Spectronaut or Diane or something else. You can download those onto that desktop PC, run those analyses on that local PC. And the Ardia system is a generic file server. I mean, it handles Proteome Discover results, raw files and sequences, especially because they're our native file formats. But you could copy any result file, any kind of data file you want on that PC and share it just like you would do with OneDrive. So let me end it there because we're out of time. But if there's time for questions, I'd be happy to answer.
Thank you. I've got a question about Chimerys. Actually, a couple of questions. Is it using MS1 traces at all in the identification, or just a possible mass within the DI window?
It's using by default MS2 traces. You can choose to do MS1 by using the standard label-free quantification method. So we'll go back and calculate XICs for the individual precursors and map those to the Chimerys results. But it's not doing both at the same time. It's only doing one or the other.
But it's not using for the identification. It's not using MS1.
It's not using MS1 for identification. It's
Okay.
pretty much all MS2 based.
Okay. I'm gonna ask one more question, then I'll let somebody else. In the Inferis list of modifications, are all of those available through Chimerys, or is that are two independent lists?
They're the same. So Inferis is used in Chimerys to do the predictions of those spectra. So the phosphorylation, the TMT, TMT Pro, oxidized methionine, carbaminomethyl are what's available in Proteome Discover 3.1. But we will have more and more modifications as we go forward. Because it's a deep learning algorithm, we need either synthetic peptide spectra data or actually very clean DDA data to use as inputs into the training to add more modification. And it gets more complex as we add more PTMs to that. But for the next release, we will have more modifications added to that list.
Thank you.
I have one question. When you communicate the Ardia with the PC host or the master spec, and you say that you can run, if I understand correctly, you can run a big project, it cannot affect the performance of the computer during the real-time analysis or transfer to the PC host? I mean,
it does.
Because...
Sorry, please continue. Sorry.
No. That's my question, because in general, what I encounter for when I'm moving, I have internet double hook connection, but when I'm moving data, you try to be sure that your instrument is not the acquisition. I know maybe.
Yeah. The upload of the data is very quick from the instrument to the Ardia server. So we've done some substantial load tests of having many systems transferring data to and from the Ardia server at the same time, and we haven't run into any issues where there was any interference. Now, all the data processing, there's no data processing actually happening on the acquisition PC. The only thing that happens on the acquisition PC is it uploads the file to the Ardia system, which is its own tower. And then that transfers the file and the processing method to another data analysis PC that's running Proteome Discover run queue. So most of the work in a workflow is actually happening on a completely separate Proteome Discover PC, except for the Chimerys part, which again is on the Ardia server. So we've tested it to be able, the Proteome Discover PC and the Ardia server can handle up to 20 concurrent analyses at a time, and you can cap it at that point. So at no point are we interrupting acquisition at all. Everything that happens is completely off the data analysis PC, and the Proteome Discover PC can handle pretty substantial load.
Well, no, really my concern is when you are moving data, not really, because I am doing another computer analysis. I never do the analysis during the data acquisition. My concern, if you are moving data in real time during the time that you do an acquisition in the PC host, you cannot affect the acquisition of the next bunch of
Yeah, we haven't seen that. For as far as we've seen and we've tested it a lot, is the data transfer is not interfering.
And I have two more commercial questions.
Mhm.
One is, if you wanted to put Chimerys and you have an old version, look like PD 2.5, you need to upgrade to 3.1 to then.
You would need to upgrade to 3.1.
But it's less expensive than buy the whole program new, correct?
Yes. Yeah, the upgrades are less expensive.
And
So
the
if you
last
had the
question-
Ardia, if you had the
go ahead.
Ardia server version and you wanted to use the Chimerys and Ardia server version, you'd buy the basic Proteome Discover upgrade part.
And the last question is, I understand the Chimerys is a contract for a license of 1 year only, correct? You need to
The cloud one is the one on the Ardia server is permanent.
It's by year, full license.
For the cloud version, you have to do it by year because the cloud cost, the cloud running data analysis on the cloud has a recurring cost to it. But if you get the Ardia server version with Chimerys, there's no yearly charge. Like you get Chimerys permanently. Like so you don't have to keep paying year by year to use the Ardia server version of Chimerys. That's a permanent license.
Finally, if I don't have the Ardia, I wanted to renewal each year. Do you have an idea how much I need to spend each year for renewal the Chimerys?
You need to talk to your sales, who's your local sales to get that.
Hi, David. This is Andy. Hi. Thanks for all that fantastic information. So I just had a quick question about. So I'll preface this by saying that I hate that I'm even asking this because I'm not, a numbers guy, and I believe that if I identify 10 proteins and one of them is the right answer to give to my client, then I don't care that somebody else identified 10,000, right? So I will say that, but unfortunately, the world we live in, when you're justifying these and you're looking at these things are sort of how they work, and I just wanted to. Can you comment on, because it seemed like from the first presentation there were a couple of slides that showed some data sets that showed both Diane, Spectronaut, and Chimerys, and it seemed to me quickly looking at those that consistently the Chimerys was getting lower protein IDs, lower peptide IDs, and a little higher CV. Is that something you can comment on? And then is that something that is expected to improve over time with these deep learning algorithm, or just sort of wanted to get your thoughts on that?
Yeah, putting me on the spot.
Sorry, like I said, if the answers are right, I don't care that it's less.
No, you're right. I'm just joking. This version of Chimerys and PD 3.1, it's a completely different technique that's being used in Spectronaut or Diane or most other kinds of software. And so this is our 1.0 release. So generally for a data set, you will get more IDs from Spectronaut and Diane than you will get for Chimerys. There are some interesting discussions to be had about what the FDR means in DIA analysis in general. Not some other software. I don't want to point anything out specifically, but we do find that some other software does have some issues with FDR control. But back to the Chimerys side, we are making improvements now. So we are going to be constantly improving the sensitivity, the throughput of the algorithm, and the precision of the measurement as we go forward. So this is our first release. We'll get better.
Yeah, no, that's great. And do you have any general concept right now about the sort of the overlap? I mean, I know as someone who's new to DIA here, and that we've been looking extensively at Diane and developing some of our own software as well as Spectronaut, that even between Diane and Spectronaut, when the overlap is not just the fact that one gets maybe 500 more than the other. The differences is sometimes well, not sometimes. It is super concerning to me. I mean, as someone who's old school MSMS DDA, it's where have you guys sort of evaluate where Chimerys sort of fits into that niche in terms of just Spectronaut and Diane as to what sort of overlap with the numbers that you're seeing?
Yeah, I guess it would be similar, in terms of there are going to be some unique things that Chimerys finds that the other approaches don't. Even if you run DDA algorithms on the same data set, you see that there's still 5 to 10% different between those, just based on some of the assumptions that the DDA search engines themselves do. So if you put a Venn diagram of the three of them, you will see some unique features on the Chimera side too.
Yeah, and one thing to add for the plasma data, we actually had searched it with Diane, with a Spectronaut, and also Chimerys, and also Encyclopedia, and MS Fragger, DIA, right? It was really interesting, and we can share the data because the application lab has that data if you're interested. We did the same overlap analysis that you mentioned, right? When it comes to the peptide ID, the numbers are much closer, right, between these approaches. But when it comes to the protein IDs, then the number are different. With the Encyclopedia and MS Fragger having the lower IDs, right, and then Chimerys, and then highest number of IDs comes from Diane. But it's really interesting that at the peptide level, the numbers are really similar, which is kind of, we felt kind of safe about the high number of IDs what we are seeing. I said, okay, this might be maybe a peptide to protein rollout, and different software do it differently. With the Chimerys, that they have a control over FDR, that's why the number is lower. But even Encyclopedia, it even is lower than Chimerys, right? But on the peptide level, like Biognosys software, Astronaut, and then Spectronaut, and Diane, and Chimerys, they were really similar on the peptide level IDs. I think the application, we presented that data at ASMS last year, but if you're interested, we can share it with you too.
I have a question. For the Ardia workflow, can you add custom components to that? Like, say, if I wanted to automate uploading to a repository?
I'm trying to think of how. It can't be on the Ardia system itself. It's not customizable in that way. But what you could do, if you, trying to think exactly how that works. I know that. I guess at a minimum, what you would need to do is use that data sync algorithm that pulls down files from the specific folder that's on the Ardia server, and then you could do whatever you want with those. But I don't think there's a way in Ardia right now to have it transfer elsewhere to a specific repository. Is this a local like NAS storage, or what kind of repository did you have in mind?
Well, I was just thinking in general, like with all of our NIH data management plans, we have to think about how to deposit the data. And that was just an example, but it could be any kind of custom component.
Yeah, I think the Ardia server itself should conform to those NIH data management plans, I think. I mean, the idea is you're supposed to store that data for several years, right? After you acquired it. And one of the goals of the Ardia server itself is to be its own repository. So you can certainly back that up, and you can archive and move data files off that Ardia server, and you can transfer it using the Ardia data sync. But what we're trying to do with that very high capacity server is to be able to keep your data around for several years on premise, and help you manage and keep your data in place that way.
Any other questions?
I've got one. The 2 Thompson window seems to, be a pretty good fit with as fast as the Astral can scan. But have you looked at optimized windows for Chimerys, and how wide can you go before you see the performance fall off, I guess? And a related question, how does it handle edge effects of splitting peptide isotopic clusters?
I guess, Khatereh, do you want to answer the isolation window one?
I think it's toward the data analysis, how Chimerys handles it. But when it comes to quantification accuracy, the best data that we have got was with the 3 Thompson M/Zs. And we had done like a 2 through 12, and and the 3 was the one that we got the best quantification data. But how Chimerys manage like a wider window versus narrower window?
Well, no, I think
maybe-
it's isotope cluster is kind of on the border, so you'll detect it in 2 different sets of cycles. So we just integrate the MS2 information for the different cycles together. So we group all that information together. So it's okay if we identify peptides on the border of the isolation window. We're taking that into account.
Okay, thank you. Any other questions? Well, David, I'm sorry, I didn't even get your name. I didn't know there were 2 speakers. I must have missed an email or something, but and Khatereh, thank you guys so much for your presentation today. We appreciate it.